All right, so we're diving into some really cool research today on DNA methylation clocks.
Yeah, it's a hot topic for sure.
It really is. I mean, we've all seen those headlines, right? Like scientists can now predict your age with your DNA.
Yeah,
Pretty catchy stuff,
definitely eye-catching.
but is it really that simple? I mean, are we really there yet? I think that's what we're going to try to find out today. And this paper that we're looking at today, I think challenges some of those assumptions.
It does. It really takes a critical look at the field.
And I know you've got a strong background, in computational biology and statistics, and I think this paper will be right up your alley.
Yeah, the methods they use are fascinating.
Yeah, and I'm hoping that you can, kind of walk us through some of that today because I think for me, I get the genetics, the medicine side of things, but the nitty-gritty of the statistics can sometimes be a bit of a black box.
Well, I'll try my best to make it clear.
Awesome. And just to make sure we're all on the same page, let's do a super quick recap of what DNA methylation actually is, right? Because this is really the foundation of these clocks.
Okay, so think of it this way. You've got your DNA right, which is like the instruction manual for your body.
Right, the blueprint.
Exactly, and methylation is like adding little notes or tags to that manual.
Okay, so not changing the instructions themselves, but kind of like highlighting certain parts.
Exactly. It's not changing the letters of the DNA code, but it can change how those instructions are read, which genes are turned on or off.
Got it.
And the really cool part, these tags, they actually change over time as we age.
That's where the clock comes in, right?
Exactly. Scientists can look at the pattern of these tags at specific spots in the DNA called CPG sites and use algorithms to predict someone's age based on that pattern.
Sounds pretty straightforward when you put it like that, but
It sounds simple, but there's a lot more to it.
yeah. And this paper that we're talking about today argues that some of these clocks, particularly the ones that rely on machine learning techniques like this thing called elastic net regression, they might not be telling us the whole story.
Right, they might be missing some crucial information about how we age and how that's connected to things like disease.
And that's where things start to get really interesting because one of the things that kind of blew my mind was this idea that the CPG sites that are the most important in these models, the ones that have the biggest impact on the age prediction, are often the ones that change the least with age.
It is a bit counterintuitive, isn't it?
Yeah, I mean, that doesn't seem to make a lot of sense at first glance.
No, it doesn't, but it all comes down to how these algorithms actually work.
Okay, so break it down for us.
Well, take the elastic net, for example.
Mm-hmm.
It's designed to find the smallest set of features, in this case the CPG sites, that can give you an accurate prediction.
So it's kind of like simplifying things as much as possible, right? Finding the bare minimum needed to make that prediction.
Exactly, and the paper argues that this can actually create a bias against CPG sites that show strong differences between healthy people and people who are experiencing age-related diseases.
Okay, I think I'm starting to see where this is going. So it's like
Yeah.
if you were trying to predict someone's age based on, how often they catch a cold.
Uh-huh.
Yeah, older people might get sick a little more often, but that's not a perfect predictor,
Mm-hmm.
right? There are other things going on. Their genetics, their lifestyle, their exposure to germs, all sorts of things that might be way more important in determining their overall health and how they age.
Exactly. And that's kind of what this paper is getting at. We need to move beyond just predicting someone's age on paper and start looking at their biological age. The age that reflects all those other factors that contribute to how our bodies actually age.
Okay. And so to do that, they propose this different approach, right? This hypothesis-driven feature selection.
Yeah.
So instead of just letting a machine learning algorithm decide which CPG sites matter, we start with a biological hypothesis about what drives aging, and then we select the features, the CPG sites that are specifically related to that hypothesis.
Exactly. It brings transparency and biological relevance back into the picture. It's not just a black box spitting out numbers anymore. You can actually understand why certain CPG sites are being included and what's driving the model's predictions.
I like that. And they actually give us a specific example, right? They developed this hypothesis-driven model that they called MFSS, which stands for Modified Forward Stepwise Selection.
Yeah, it's a mouthful.
It is a mouthful, but the important thing is that this model was still incredibly accurate at predicting age, and it worked well across different groups of people.
Yeah, and it was much simpler and easier to interpret than some of those fancier machine learning models.
Okay, so simpler, more transparent, more biologically relevant. I'm liking this approach, but what really got me thinking was this whole concept of model coherence and feature rectification.
Ah, yes, now we're getting to the really interesting stuff.
Yeah. Because remember how we were talking about how some clocks have trouble distinguishing between healthy aging and inflammaging, that aging that's driven by chronic inflammation.
Right.
This is where I think things start to get even more
They
interesting.
do, yeah. So this is where this idea of incoherence comes in. The authors point out that the direction of methylation change, like whether it goes up or down at a specific site, can sometimes actually cancel out the model's prediction.
Okay.
And this can happen even when there are big differences between the methylation patterns of healthy people and those who are, experiencing inflammaging.
So it's like you've got these CPG sites, and they're kind of like levers, right? Pushing the age prediction up or down.
hmm
But sometimes those pushes balance each other out, even if the individual changes are pretty dramatic.
Exactly. Yeah, it's a great analogy. It's like a tug of war where both sides are pulling really hard, but the rope doesn't move.
Right, it's just stuck.
And that's where this idea of feature rectification comes in. This
Mm.
is a method that makes sure that all the pulling, so to speak, is in the same direction.
Okay, so it's like we're getting everyone to pull on the same side of the rope now.
Exactly. And that gives us a much clearer signal of inflammaging. It's a really elegant way to make these linear models more sensitive to those subtle age-related changes that might otherwise get lost in the noise.
I can see how that would be super useful, especially for
Mhm.
something like inflammaging. But I also wonder if, focusing solely on getting everything to pull in the same direction
hmm
might be oversimplifying things a bit.
Yeah, that's a great point, and you've hit on a key point that the paper raises. While feature rectification is a clever solution for improving the sensitivity of linear models, it doesn't address a fundamental limitation.
And that is,
Biological aging is incredibly complex.
oh yeah, we know that.
And trying
Yeah.
to capture all of that with a simple linear model is like trying to fit a square peg into a round hole.
Yeah, I'm starting to see the problem here. So when we're talking about limitations of linear models,
hmm
can you break that down a little bit more for me? Like, why are linear models not ideal when we're talking about something as intricate as aging?
Well, linear models are great at capturing straightforward relationships where one thing directly affects another.
Right, cause and effect.
But aging isn't like it's this dynamic interplay of genetics, environment, lifestyle choices, even random chance, all weaving together in these intricate feedback
That's
loops.
like a giant messy web.
Exactly. So trying to squeeze all of that complexity into a linear model is going to leave out a lot of crucial information.
It's like trying to map a whole galaxy using only a ruler.
Exactly. You're getting it. And that's why, even though feature rectification helps us see inflammaging more clearly in a linear model, it's still a bit of a band-aid solution.
Okay.
It doesn't address the fact that we might need entirely different models to truly grasp the nonlinear nature of aging.
Okay, so are we saying we need to completely ditch linear models and go for something more complex? Like, what are the alternatives here?
It's not necessarily about abandoning linear models entirely. I mean, they still have their place, especially in the early stages of research when we're trying to establish, basic relationships. But as we gain a deeper understanding of aging, we need to be ready to embrace more sophisticated models that can reflect the true complexity of these processes.
Okay, that makes sense. So what would those more complex models look like? You were talking like serious, advanced math here.
We are talking about advanced math, but the good news is that with the incredible advancements in computational power and data analysis techniques, we're now able to develop and test these more sophisticated models.
Okay.
For example, we can now use nonlinear models that can account for those intricate feedback loops and interactions that we talked about.
So instead of just looking at like individual CPG sites and how much they change, we can start to see the bigger picture, how they all influence each other over time. That's getting me pretty excited. What kind of possibilities does this open up?
The possibilities are huge. Imagine building a model that not only predicts chronological age, but also captures how inflammation, DNA, damage, cellular senescence, all these hallmarks of aging interact with each other.
That would be incredible.
Hmm.
We could potentially use these models to identify individuals at higher risk for age-related diseases, personalized treatments,
Mm.
track how well those treatments are working. Are we talking about like truly personalized medicine based on someone's unique aging profile?
That's the ultimate goal.
Yeah.
And this paper really lays the groundwork for moving in that direction.
This is definitely a lot to unpack. So let's take a step back and just kind of recap what we've learned so far.
Sounds good. So we started by looking at the limitations of current DNA methylation clocks, especially their reliance on machine learning techniques that may overlook biologically relevant features.
Right. And we kind of jumped into this whole idea of hypothesis-driven feature selection,
Yeah.
which seems like a much more transparent and informative approach. And then we got into this really fascinating concept of model coherence and feature rectification, which can make these linear models more sensitive to inflammaging. But as we've just been discussing, we also need to acknowledge that relying solely on linear models might not be enough to capture the full picture of aging. There's this whole other level of complexity that we're only beginning to explore.
Exactly. And that brings us to a crucial question. What's next for DNA methylation clocks? Will they continue to evolve and become more sophisticated, or will they be replaced by even more powerful models of aging?
That's the big question, isn't it? No, while we can't predict the future, we can certainly explore some of the emerging trends and research directions that are shaping this field. And I'm already eager to hear, like, what's next? What exciting advancements are on the horizon for DNA methylation clocks? So much to think about. But before we get too carried away with future possibilities, let's bring it back to the paper for a sec. I mean, they really honed in on inflammaging,
Yeah.
that whole chronic inflammation thing.
They did, yeah.
And it got me thinking, I mean, what if we could apply this whole idea of feature rectification?
Hmm.
Making sure all those methylation changes are pointing in the same direction. What if we could apply that to other age-related conditions?
Yeah. Beyond just inflammation.
Right, exactly.
Yeah.
Because, I mean, there are so many other processes that are linked to aging, right? Oxidative stress, those pesky telomeres,
Yeah.
getting shorter changes in the gut microbiome.
Hmm.
Could each of those have its own unique methylation signature?
Absolutely. I mean, each of those processes leaves its mark on the epigenome, shifting those methylation patterns. And we're just starting to unravel those connections. It's really exciting.
So instead of just having like one generic biological clock, are we talking about like a whole panel of clocks fine-tuned to specific conditions?
Potentially.
Like imagine a clock that measures your cognitive age based on methylation changes in the brain, or one that predicts your risk of heart disease by looking at patterns in your blood vessels.
Exactly. And it's not just about predicting individual risk either. Think about drug development.
Oh, yeah.
If we can pinpoint CPG sites that are consistently affected by a particular disease, we could target those sites with therapies designed to modify methylation.
Oh, wow. So like epigenetic tweaking. That's getting pretty sci-fi.
It is, but it's a real possibility.
That's incredible. But hold on a sec. We've been focusing so much on what changes in DNA methylation, but what about the why? I mean, we know these patterns are influenced by a ton of factors: genes, environment, lifestyle, heck, even our thoughts and emotions.
You're right. It's the ultimate puzzle, isn't it? We know methylation changes, but what's driving those changes in the first place? That's the million-dollar question.
So instead of just using these clocks to measure biological age, could we actually use them to understand what's pushing it faster or slower?
That's the dream.
Like if someone's epigenetic age is way older than their actual age, could we pinpoint, what's contributing to that? Is it stress? Is it diet? Lack of sleep? Whatever.
Yeah. Imagine being able to say, hey, based on your methylation, it looks like stress is a major factor in your accelerated aging. Or, your epigenome suggests you might benefit from focusing on gut health or reducing inflammation.
Wow, we're talking about truly personalized medicine tailored to your unique epigenetic profile.
Yeah.
That's incredible. But to get there, we need a lot more research, right?
Absolutely. We're just scratching the surface here.
Yeah.
We need larger, more diverse data sets. We need to refine our models, move beyond those linear approaches, and really capture the full complexity of the epigenome. And we need better tools to interpret those methylation patterns and link them to specific biological processes.
So much work to be done, but so much potential. It's really exciting stuff. So for everyone listening, here's a final thought to ponder this paper we talked about today, focused on inflammaging, right? But what other factors, beyond the ones we've mentioned, might be shaping your epigenome as you age? What could those changes reveal about your health and your future? It's a whole world of discovery hidden within these methylation patterns. So keep exploring, keep asking those questions. And who knows, maybe you'll be the one to unlock the next breakthrough in the science of aging. Thanks for joining us on this deep dive. Until next time, stay curious.